Right, up next, we are going to hear from Brenna Friday, who's been working on a project this summer with Mike Fraker. All right, great. We can see your slides, Brenna, but I also see your icons. There we go.
Okay, is this better?
That's great.
Okay.
Are you going to use your webcam?
Oh, yes, sorry.
We don't see you yet, no problem.
Okay.
Great. We can see you, and welcome, Brenna. I'll let you introduce your talk.
Okay. Just to be clear, you guys can't see my little webcam in the corner?
Yeah, we can hear you and see you just perfectly. Your slides are coming through, so you're good to go.
Okay, good. Hi, everyone. I'm Brenna. This summer, I worked with Dr. Mike Fraker on a project called Understanding Biological Changes over Spatial and Temporal Gradients from the Edwin S. George Reserve. So for those of you that aren't familiar, the Edwin S. George Reserve sits a little northwest of Ann Arbor. It's been protected since 1930 and is completely a research area. There's no public access. This makes it a really wonderful place to study all of the marshes, swamps, and bogs and ponds that can be seen on this map that are housed within the reserve bounds. So the data that I worked with this summer came from a project designed by Dr. Earl Warner from the University of Michigan. He collected data from 37 ponds over a 15-year survey using methods such as pipe sampling, dip netting, and seining to collect over 160 taxa of amphibians, fish, and macroinvertebrates. So he also collected a lot of environmental data on hydroperiod and bathymetry mapping, which contributed a really wonderful picture of what was going on in the overall reserve. So when I was given this data at the beginning of the summer, Dr. Fraker and I kind of tried to come up with some way to evaluate it all to understand what it can tell us. So we came up with two questions, one being what methods should be used to evaluate changes across different biological, spatial, and environmental scales, and using some kind of case study, how did two distinct frog species respond to environmental changes over this time period? So the two frog species that I'm going to talk about today are Rana calamitans, the green frog, and Rana sylvatica, the wood frog. So these two species are pretty closely related, but they have very different life histories. Green frogs breed very late in the summer. I actually heard one calling last week, if anybody's been listening for them. Whereas wood frogs are usually the first frogs that you hear calling in March, so they breed very early in the spring. It also takes them a highly variable amount of time to metamorphose from a newly hatched tadpole into the terrestrial frog that we see in the picture here. So for green frogs, that process can take anywhere from 3 to 22 months, whereas wood frogs are very speedy and they develop in 2 to 4 months. This timing really determines what kind of habitat preferences that the two species have. Green frogs prefer to lay their eggs in permanent ponds, which makes sense so that their tadpoles aren't developing in ponds that are drying up around them. Whereas wood frogs don't really have to worry about that so much. Their tadpoles develop so quickly that by the time they can lay their eggs in ephemeral pools, by the time the ephemeral pools are dried up, they're totally developed and are totally fine. So that's all of the background I'm going to get into for now. I'm going to touch on some of the preliminary results that the analysis gave moving forward. So this is a timeline of particular standout environmental events that I noticed over the course of the sampling period. So we didn't see any huge changes overarching changes, I should say, in temperature, precipitation, but that was expected given such a short window in terms of only 15 years. I would like to call your attention to some of the more extreme years, you could call them, in terms of our sampling period. We saw generally that our driest and our hottest years fell between 1998 and 2003, whereas the wetter and cooler years fell in 2008 and 2009, respectively. So this is something that I'll be talking about as the presentation goes on, but I think it's helpful for you to just see this and try and keep this pattern in the back of your head as I move forward. So given the changing environmental conditions from year to year, we also wanted to evaluate how the frogs were distributed across the ponds in the reserve over time. So I created this map. It's animated, so show how the distribution looked each year, but I want to draw your attention to the legend on the left first. So the red ponds that you'll see represent ponds in which Brenna sylvatica species were only found, those wood frogs. Green ponds represent ponds in which we found both species. Blue ponds are ponds where we found exclusively Brenna calamitans, or green frogs. And gray ponds represent ponds where we didn't find either of the two species. So I'm going to play this, if I can find my cursor. Oh. Uh-oh. For whatever reason, I can't click to play my video.
Brenna, you might be able to do it if you want. You could exit your presentation mode. It might work. I don't know.
Okay.
I'm not an expert in showing videos, but we can still see what's going on.
That's okay. If you guys can still see my screen, I'm gonna just play it from here and talk a little bit. So as it animates, you can see that at the beginning of the time period, we're seeing a large number of those red ponds, particularly in this year. The following year, we have a lot of ponds that are unoccupied. It was a very dry year, but those red ponds continue to increase in number until we reach later in the time period, about 2006, 2007, we see an increase in the number of green ponds that show up on the map. Okay, so I'll go back into this. And now these are the same data represented in just a different way. On the y-axis, we have the number of ponds occupied, and the x is each year. All of the colors represent the same group, so the legend still applies. But as you may have mentioned in the animated, or as you may have seen in the animated maps, the number of ponds that we found exclusively wood frogs normally outnumbered the other types of ponds, especially at the beginning of the sampling period.
Brenna, I'm gonna interrupt you really quickly. We're not seeing your graph. We still see the map, the color-coded map.
Is anything changing now?
No.
Interesting.
Now we see a change, but we see a slide that says not all your pond. There we go. Okay. There's your figure. Sorry if I made you lose your train of thought.
No, that's okay. The joys of, okay, we're good. Sorry, I just want to exit this now. Okay, redo that. So we see that the number of ponds with wood frogs does change over the course of the time period, the sampling period. They see some declines in years like 2000 and 2003 and 2004, which were a part of that particularly dry period. But we see that the wood frogs do seem to rebound, and they increase in their number of ponds occupied after those times. The green frogs don't seem to take such a hard hit during those drier years. And what's interesting with that is that we also see towards the end of the sampling period, when we enter into those wetter, cooler years, we start to see an increase in the number of ponds occupied by green frogs and an increase in the number of ponds occupied by both species. By 2009, these ponds actually outnumber those occupied by exclusively wood frogs. And in 2010, we see some overlap there. Something to consider that I haven't really talked about is the fact that not all of these ponds are full year round. So when you look at the number of ponds occupied, you really should be considering which number of ponds provide valuable habitat to both of the frog species every year. In order to do this, you have to consider hydroperiod. So I created another map that shows the different groups of ponds based on how often they dried during our sampling period. You can see we found 6 ephemeral ponds that were dry for the majority of the sampling period. They're shown in red here. We found 15 semi-permanent ponds that were dry for some of the sampling period, and 14 permanent ponds that were dry for even less of the sampling period, less than 25%. We found, when you look at how many wood frogs and green frogs are actually found in these ponds, there were significantly fewer frogs found in the ephemeral pools versus the semi-permanent and the permanent pools. So this is to be expected, given the habitat preferences that I talked about, but it's also important to note that the 6 ephemeral pools were vastly outnumbered by the number of semi-permanent and permanent ponds, that were found in the reserve. When you analyze these data sort of from a different lens, we see that the population distribution for both the species across hydroperiod changes. So over time, we saw that green frogs on the left the majority of their population was found in permanent ponds across the board, especially during those particularly dry years from 1999 to 2003. They were exclusively found in permanent ponds. Outside of those years, there were some that ventured into ephemeral ponds and semi-permanent ponds, but the majority were found in the permanent, which is what the literature suggests. On the other hand, the wood frogs, we expected to see that they were found mostly in ephemeral ponds, and we do see that some of the population is consistently living in ephemeral ponds, but never the majority. This is likely due to the same reasons that the green frogs were moving around, again, that change in dry years versus wet years. In dry years like 2000, 2003, 2004, we see no wood frogs in ephemeral pools, likely because that habitat just was dry that year and wasn't really valuable to them. But in the wetter, cooler years in the later sampling period, we see more frogs showing up in the ephemeral ponds. There are a couple of reasons as well as to why the majority of the frogs across the board are seen in the semi-permanent ponds. We think it's likely that recruitment is higher in these ponds for wood frogs. And also, it's potentially due to the fact that wood frogs have very high site fidelity rates. So they will return to the ponds in which they were hatched to lay their own eggs when they're sexually mature. So that can cause some delay in how many ephemeral ponds are occupied and what % of the population we find there over time. So given all these data, we were able to kind of identify some trends, and I think that maybe some climatic factors were playing into how the population distribution is fluctuating from year to year, but we were not able to totally pin down what was driving them. In order to answer this question, we turned to the tool called gradient forest. So gradient forest analysis is very similar to random forest, which some of you may be more familiar with, but it builds on that and evaluates changes over a community level. What it does is that it identifies the best forest of regression trees to explain variation in our data. So the way that we went about it is that we input our data in two ways. The example that I'm going to talk to you about right now just used data from the semi-permanent ponds, those 14 semi-permanent ponds. So we took all of the data from all of the species that were collected in those semi-permanent ponds across all of the sampling years, calculated their means and then z-scores, and put them into the program. The reason that we use z-scores was to make the data comparable across different dimensions. We did the same for the environmental data. So we had 9 environmental predictors from semi-permanent ponds across all of those sampling years, and we input those data into the program as environmental predictors. One of the first outputs can be seen here on the graph. What is listed here are our 9 environmental predictors ranked from most important to least important. So the ones that are contributing the most to the model, to the variation in what we're seeing, in R-squared weighted importance, to the least. You can see here that the identified most important predictor is the total number of dry days that were identified. Gradient forest takes this one step further. It can actually not only identify the most important environmental predictors, but also the most vulnerable species to changes in those environmental predictors. So again, this is the analysis of the semi-permanent ponds at a species level. On the graph here, you can see that z-scores along the x-axis to represent the changes that we saw in our data. The y-axis represents cumulative importance value, which is how much a predictor contributes to the goodness of fit of the model for each particular species. The top 5 most vulnerable species are listed on the legend on the top left. And you can see that to continue the narrative, our green frogs are listed as the top 2 sort of most vulnerable species to this change. So they're listed in the hot pink line and the blue line that you can see. When we see the data and we examine, we try to find areas of change, it seems that between 0.5 and 1 z-score, there's a jump in that cumulative importance value. What that tells us and how we should interpret this is that the groups of green frog population and potential density below that value differ from the groups above that value. This does not tell us any kind of directional change. It doesn't indicate exactly if this is an increase or a decrease, but in order to evaluate what kind of change this truly is, we have to go back and analyze our raw data before gradient forest analysis. So here we have that plot that I just showed on the left, but the new plot that you're seeing is our calamitins density, so the larva per meter squared, plotted against total number of dry days on the x-axis. When you look to see exactly where this threshold is on the plot on the left, you see it falls at about 0.75 z-scores, or 0.75 standard deviations above the mean. When you calculate what that value would be for total number of dry days, it comes out to be about 124 dry days. That value is represented by the dotted line on the plot. What's interesting to note is that every point to the left of that line, so any number of dry days that is fewer than 124, shows some kind of green frog density. So what we see before that point is a presence of green frogs. What we see after that line is that all of the density values drop to 0. So after that line, we are unable to find any green frogs in the semi-permanent ponds. What this should be taken as is less of a line, we think, and more of a spectrum around that area. Given more data, we could really say that there is a range, likely around that point, in which density values start to decline based on total number of dry days. We feel like this tool could have major implications for future research. So this quote comes from the Climate Change Adaptation Plan for Coastal and Inland Wetlands in the state of Michigan from 2012. And it states that it will require extensive annual monitoring and observation of changes in wetlands to fully identify which wetlands and wetland-dependent species are most at risk to changes that will occur as a result of climate change. Using gradient forests as a tool, it can help us address a lot of this sort of requirement, because it can address not only identify the most important environmental predictors for certain wetlands, but it will also help us identify the most vulnerable species to changes in those environmental predictors. So we hope that this tool can be used as analysis of climate change in the Great Lakes ecosystem. And also we feel that the data that's collected from the Edwin Storage Reserve can be applied in a broader scope to more coastal and in the wetlands in the Great Lakes. So in conclusion, long-term monitoring programs like this help establish environmental context for biological change. Different species' presence in and preference for certain habitats is very dynamic, as we saw with the wood frogs and how their preference for ephemeral pools changed over time. Hydroperiod is a significant predictor for overall frog density in R. clamitans and R. sylvatica. And gradient forest is a useful tool in evaluating these changes across spatial, environmental, and biological gradients. But correctly evaluating outputs should be navigated with caution and an abundance of data. So I wanted to say thank you for all of my mentors in this program. Dr. Mike Fraker has been such a big help. And I also wanted to give a certain shout out to Chris Davis and the rest of the ESGR survey team that collected all of this data for 15 years. Thank you.
Fantastic job, Brenna. Thank you so much. Really interesting study and results. Let's see who has some questions for you. If you've got a question, feel free to raise your hand or type it in the chat box. Just wait a few seconds here. Okay, I'm not seeing anything come up. If there's something, the fellows will be on the webinar for the whole time this morning. So if there's something that comes to someone's mind, or if I'm switching off too quickly, feel free to go ahead and add that in the chat, and we can come to it at the end, or if there's time after another talk. Thank you very much, Brenna. Really enjoyed that. I think we all did, and great job.